Hi everyone, today we're going to have a look at some of the key topics and a lot of foundation knowledge related to our genetics unit. We're going to have a look at genes and alleles, how they're passed on. We're going to have a look at some of Mendel's laws, and we're going to have a good look at an example that I've put together here in the center at the very end. To start off with though, let's have a bit of a recap and let's take it right back to the very beginning. We are going to look at the ways in which organisms can reproduce. So organisms are living things, and we know that they can reproduce in two ways. So they can either reproduce asexually or sexually. If they reproduce asexually, these organisms have one set of chromosomes. So that would be one set of chromosomes and one of each gene, and they reproduce by the process of mitosis. So their offspring are identical to themselves, and there's little to no variation between parent and offspring. So asexually, one set of chromosomes reproduce via mitosis. Sexually reproducing organisms require two different organisms to come together. They have homologous chromosomes, which means that they have pairs of chromosomes with the same genes. So homologous chromosomes mean that they are the same chromosome. So each chromosome will have the exact same gene on it. However, one chromosome will come from mum and one chromosome will come from dad. Before we go any further, I want to quickly give you a little note for a definition of genes. Genes are segments of DNA that have a function. In that sense, it means that they code for something meaningful. Either they're going to produce a protein or they form part of the production of a protein, or they have a role or a job in the body. So these are very special parts of DNA that actually code for something specific. Now, these pairs of chromosomes that we've just been talking about, the homologous pairs of chromosomes, they are chromosomes with the same genes on them, meaning that they have the same type of information on each one of them. We know that one chromosome comes from mum and one chromosome comes from dad. So if I drew these chromosomes, and remember chromosomes are DNA that are tightly wound and wound and wound and wound and wound, and the DNA strands, if we were to unwind them, would have all the nucleotides together here and all the nucleotides together here. Now I've told you that these chromosomes here are homologous, one from mum, one from dad. I've then unwound it and I've had a look at the DNA sequence that is relating to one specific gene. Now this gene is the same gene on each of these. This gene is the same gene as this one, only this one belongs to the mother's chromosome and this one belongs to the father's chromosome. So the first thing you should see is that you should have a look at and go, okay, well, the nucleotide sequence or the DNA sequence is different in both. And that's perfectly fine. What it means is that this DNA sequence and this DNA sequence are sequences that relate to the same trait. However, the sequence that mum's given sequences for a different outcome or a different version of the trait, whereas dad's sequence is for a different version of the trait. So they each have their own sequences that go towards providing information for the same gene. This gene could be related to hair color, eye color, height, or just about anything. So in a quick example, we might say that this gene, this red gene that I've identified on the chromosome, might relate to hair color. And the gene that mum has passed on is for blonde, and the gene that dad has passed on is for brown. The gene is still the hair color gene, but the one that mum's passing on and the one that dad's passing on is different. So homologous chromosomes, the exact same genes, so both hair color genes on each one, and these homologous chromosomes have their own set of genetic information or coding for that particular gene. Something else I'm going to quickly add on here is that along these chromosomes there are many, many, many genes that can be identified. And they are all along the chromosomes, and there are many, many, many more genes than there are chromosomes in a cell. The genes that are along one chromosome are said to be gene linked. So that's the relationship that they have. I'm going to make that slightly darker for you so that you can see that on your screen. because that's one of our key words for today. So gene linked, genes on the same chromosome. Okay, so gene linked. That's fairly important because if you're thinking about it, if each chromosome, one from mum and one from dad, have the exact same genes on them, then there's got to be a link between them. So they're completely independent chromosomes that have come together in the one organism together, but we know that we'll be able to find a matching chromosome, a homologous chromosome inside the cell. Okay, so the sequence is one gene, so one type of gene, and the DNA sequence on the individual chromosome is unique. There are many sequences, many sequence alternatives for each gene, and these are called alleles. So these sequences here are just two of the sequences that are alternatives for these genes. So there are many, many alternatives for these specific, for this specific code that will fit for that gene. We call these alternatives alleles. So what that means is, and I'm going to grab my pen back again. What that means is that these guys here are alleles. That's one allele and that's another allele. So in sexually reproducing organisms, they have two alleles for every gene. Two alleles for every gene. If we want to take it right back to our asexually reproducing organisms, these guys here have one set of chromosomes, which means they will have one allele for every gene, or their alleles are their genes. If we're looking forward, so these are called alleles. Alleles are represented by a letter. Alleles can be dominant or recessive. Now, dominant alleles or dominant sequences override the code of the partner allele. So we know that there are going to be two alleles that are given, one from mum and one from dad. If one of those alleles is a dominant gene or a dominant allele, it will override the other one, and so what is expressed will actually be the code from the dominant allele. To put it simply, if mum gave a blonde gene and dad gave a brunette gene, we might say that brown is dominant to blonde, and so whilst the offspring will have a blonde and a brown gene, the offspring will have brown hair because it's dominant. So the dominant allele overrides the code of its partner allele and is represented by an uppercase letter. So if we were going to give anything that is a dominant gene or a dominant allele, will be represented by an uppercase letter. If we're looking at a recessive gene, however, recessive genes are overridden easily. We usually need, well, not usually, we do. We need two recessive alleles in order for that gene to be expressed, and they're represented by a lowercase letter. So if we go back to our situation up here where mum's handed over a blonde allele and dad's handed over a brown allele, if the children were going to be offspring, the offspring were going to have blonde hair, we would need to get two blonde alleles, one from mum and one from dad, in order for the offspring to be blonde. And we would represent those ones with lowercase letters. So if we had blonde, we might use the letter B, and you can choose the letter that you use. We would have a lowercase b and a lowercase b, which would provide us with an offspring of blonde hair. If dad provided a brunette or a brown offspring, a brown allele, it would join with the blonde allele from mum, and we would have a dominant allele. So we would have an uppercase B and a lowercase b, is what we would get, and the offspring would have brown hair because brown would be dominant in this situation. So this is where we're going to start talking about those combinations. Alleles, the combinations of alleles, mums and dads alleles, determine the characteristics that are shown. So combinations can be called a few different things. They are either homozygous or heterozygous. Homozygous means that both alleles are the same. Homo meaning same, so both alleles are the same. For example, if we have a look over here, we've got our homozygous dominant, which means we have two capital letters, and so two dominant alleles that have been passed down, or homozygous recessive, which means we'll have 2 lowercase alleles being passed down. If we applied it to our situation we've been talking about over here, in this situation, if the big capital B was standing for brown and the lowercase b was standing for blonde, this organism will have brown hair, whereas this organism will have blonde hair. In the situation over here where we have a heterozygous, hetero meaning different, different alleles, so one dominant and one recessive, we would have an offspring that will show the dominant allele of brown because it's dominant and it will override the recessive gene or the recessive allele. Okay, allele letter combinations are known as genotypes. So what that means is that these combinations here of big B, little b, and big B, little b, these are called genotypes. All right, so genotypes are the allele combinations represented as letters. The traits expressed by the organism are called phenotypes. So phenotypes are what we actually see. So for example, this one here might be brown, this one here blonde, and this one here brown. So it's the characteristic that is actually expressed by that organism. Before we go on to our example that we have over here, I'm going to quickly draw your minds back to when we talked about meiosis and crossing over and its value. And we need to, because when we're working out what the chances are of an offspring possessing a specific trait, we need to think back to not what the parents are actually donating to the offspring, but more often than not what their parents have donated to the parents of the offspring. So I'm going to talk to you about one of Mendel's laws, which is the law of segregation. And what it is that the genes in gametes contain only one of the alleles for each gene, meaning that they segregate after crossing over. So we've spoken about this before. I'm going to grab my texters back. Just 1 second. That's right. And they're all still crossed over. So in our video where we had our meiosis conversation, and even when we've talked about gametogenesis, what we have talked about is the process of crossing over and how it occurs. So if we had, and I'll grab out just 2 of these now. If we have our 2 homologous chromosomes, meaning that they have the exact same sets of genes on them, but they have different alleles on them for those genes, and it's going through the process of meiosis to make gametes, because we're talking about the formation of gametes. In meiosis 1 and in prophase 1, before the chromosomes are actually pulled apart and the whole process begins, they go through that process of crossing over, where the alleles that have come from mum and the alleles that have come from dad of the organism that is creating the gametes are swapped around. It's basically a reshuffling of the cards. So when they're swapped around, parts of the genes, so the alleles, some alleles will jump from one chromosome to the other. And then when those chromosomes are then pulled apart to make an individual gamete, remember, gametes are only allowed to have one chromosome in them, so that when they combine with the egg or the sperm, they create a full set. They have completely different, they offer completely different combinations of alleles and genes. So the law of segregation states that only one of the alleles, so that means just one of the alleles, is able to go, is inside of one gamete. What that means is that if this allele that we swapped over at the beginning, and I'll put it back to the way it was, if this allele that we swapped over at the beginning, if this was dad's gene and it was a brunette or a brown, and this one was mum's gene and it was a blonde, and this gene down the bottom here, let's say that it's brown eyes, and this, let's go with this. Let's go with brown and brown eyes and blonde and blue eyes. What that would mean is that during the process of crossing over, these alleles are swapped, and so this chromosome now offers brown eyes and blonde hair, while this chromosome now offers brown hair and blue eyes. And so when they're pulled apart, despite the fact that they have they've crossed over, only this allele here, or this chromosome here, still only has one eye color and one hair color allele, and one eye color and one hair color allele. So the law of segregation says that each gamete only contains one allele for each gene. It doesn't end up with two alleles, one allele for each gene. Later on down the track, when it combines with the sperm or the egg, it will end up with two alleles. But in the process of creating a gamete, one allele per gamete. The second one is the law of independent assortment. All alleles are recombined during crossing over so that the gametes have a combination of alleles from their parents. Basically, what this means is that the cards can be completely shuffled. So the alleles have basically an infinite possible assortment or arrangement. So we have a great variation that is provided by this law occurring. So during that crossing over period, there's not just two alleles on each of these chromosomes. There are many. And so some will jump across, some will stay, and that ratio of staying and crossing will change across every single one of them that is created. So the assortment, the recombination provides a massive rate of variation, and this massive rate of variation increases the species survival greatly, because with more variation and more variety, there's a lot more that natural selection has to work with in order to increase the species' chance of survival, regardless of what's thrown at it. Okay, so let's have a look at our example now that we've covered all the content that goes around it. We're going to use this scenario. So we're going to look at eye color, and we're going to talk about brown and blue eyes. We're going to keep it really simple, and we're going to link this to a complete dominance situation. So this might not mean too much to you now, but what it means is that we have one dominant gene and one recessive gene. There are other situations, we'll cover them later, but for us to go through this, we're going to talk about complete dominance. Okay, so eye color, we've got complete dominance. I'm telling you that we're going to use the letter B to represent the gene of eye color, and we're going to use capital B to represent the allele of brown, and because it's dominant, and a lowercase b to represent the allele of blue because it is recessive. So we have a mother and a father. We have the mother who has a big B and a little b, and therefore is heterozygous because they're different alleles, and her eye color is brown because the brown allele will dominate the blue allele. So we have a mother, heterozygous, and brown. We also have a father who has two little b's and therefore is homozygous, and he's homozygous recessive blue. He could be homozygous dominant brown, but because they're both little b's, he's homozygous recessive blue. So what we're going to do is we're going to use a Punnett square, which is what we call these, to work out what variations and what combinations the offspring could have. So across the top of it, I'm going to write the mother's genotype. Remember, the combination is called a genotype. So mum has a big B, little b, and dad, I'm going to write him down the side, two little b's, little b and little b. In the middle, I'm going to write out the combinations. Now, if I have a dominant allele, I'm going to write it first. It makes it easier to work out what my phenotypes are going to be later. So capital B, little b, little b, little b, big B, little b, little b, and little b. Okay, so I have two matching homozygous offspring, and I have two recessive heterozygous offspring and two recessive homozygous offspring. So I'm going to write out my genotype ratios first. So this is where we work out what portion of the offspring are likely to have a particular type of genotype. So if I look on this, I've only got two types of genotypes. So I've got a big B and a little b, and I have a little b and a little b. So my ratio is, I've got 2 of these and 2 of these, so that would be 2:2. But if I simplify that down, it is the same as 1:1. I can also write that as a percentage, and so I would say that I have 50% is to 50%. So we can represent this in a few different ways. The next one we need to be able to calculate or to write or record is the phenotype ratio. Now, phenotype ratios are what is expressed. So in this situation, we're looking at eye color. What eye color do these offspring express? So I'm going to use, instead of going back to the Punnett square, I'm actually going to use the genotype ratio that I've looked at and work out what color these have instead of identifying each square and then recalculating. So if I have a big B, little b, it's a heterozygous and it's brown because brown is dominant. So I'm going to have a phenotype of brown. And then this one over here, I've got a homozygous blue. So the phenotype that will be expressed is blue. This one here is going to be blue because there is no brown allele to dominate it. So 2 different phenotypes. In my ratio, it's going to be the same as the genotype ratio because I have 2 browns and 2 blues. So I'm going to have 1:1 and 50% is to 50%. In this situation, it is possible for you to end up with fewer phenotypes than it is genotypes, because remember, I could have a homozygous brown here as well, but when it comes down to the phenotype ratio, those 2 will combine because they will both express a brown gene. Hope you've got it. Good luck.